which is entomology. Whether it's protecting our crops, understanding complex ecosystems, or managing disease, entomology is the vital discipline that helps us actually understand our six-legged neighbors. So our explainer today acts as your training manual. We're going to cover the science of insects, the head and antenna types, the thorax with its legs and wings, how anatomy becomes taxonomy, the field's guide, and finally, why entomology matters.
All right, part 1, the science of insects. Let's see how this builds from the ground up, starting with what actually makes an insect an insect.
The golden rule of class Insecta comes down to a fundamental three-part body plan. Regardless of the species, they all possess a hardened external skeleton divided into three very distinct regions. You've got the head at the front, the thorax right in the middle, and the abdomen at the rear. Basically, if a creature doesn't have this specific segmented layout, it's not a true insect. Moving to part 2, the head and antenna types. Zooming in on the very front region, the head houses the insect's primary sensory tools. This includes their compound eyes, their uniquely adapted mouthparts for chewing or piercing, and, most importantly for our focus today, their antennae.
Check out the sheer, incredible diversity of these jointed feelers. Insects rely heavily on them to detect odor, sound, and taste. But for entomologists, pairing these distinct shapes with their scientific names makes identification incredibly easy. You've got the thread-like filiform antennae, the beautiful feather-like plumose, the bristle-like cetaceous, and bipycnate, which literally look like tiny combs. Recognizing these specific shapes is step 1 in a field entomologist's training.
Section 3. The thorax, legs and wings. Moving right along to the middle section. If the head is the sensory center, the thorax is essentially the powerhouse engine for the insect's movement. It's exactly where the legs and the wings attach.
Now, these three pairs of segmented legs are highly adapted survival tools. They aren't just generic limbs. Take the diving beetle. It has legs specifically flattened and fringed with hair for swimming. A flea or a grasshopper? They have incredibly muscular legs built purely for jumping. And a honeybee has legs equipped with specialized structures perfectly built for carrying pollen. For a scientist, looking at the shape of these legs is an instant clue to how the insect lives and how it should be classified.
The thorax also anchors the wings, which are incredibly varied and serve as another major identifier. Some insects have thick, leathery, horny wing covers, while others have delicate, membranous wings covered in this intricate network of veins. The exact shape, size, and vein pattern act as massive clues for scientific identification.
Part 4. Anatomy becomes taxonomy. So the crucial takeaway here is that these specific anatomical features, the leg shapes, the wing structures, the antenna types, they aren't just neat bits of trivia. They are the exact, practical keys that entomologists use to name, sort, and classify insects into logical groups.
Decoding how scientific naming actually works relies heavily on the Greek suffix ptera, which simply means wings. When you know that, suddenly these complex scientific orders make perfect sense. Lepidoptera translates to scale wings, which perfectly describes butterflies and moths. Coleoptera means sheath wings, describing beetles with those hard wing covers. Diptera means two wings, which groups flies and mosquitoes. For an entomologist, tying the physical anatomy of these wings and antennae directly to their scientific names makes identifying even the most complex insects surprisingly easy and logical.
Ever since the 1700s, when Carl Linnaeus developed this biological classification system, scientists have used this precise two-part naming language to get even more specific. Take Romalia microptera, the eastern lubber grasshopper. The genus is always capitalized, acting kind of like a broad family name, while the species is lowercase, acting like a first name. This systematic tracking allows researchers to understand an insect's unique characteristics and its evolutionary lineage through DNA sequencing.
Section 5. The Field Guide. Now let's step out of the classroom and look at the hands-on work. How do professionals actually collect, preserve, and label these specimens out in the field for future research?
You see, properly preserving an insect requires strict scientific precision. Typically, an entomologist will drive a pin vertically right through the thorax or through the right wing for certain beetles. After the insect is pinned and its wings and antennae are carefully positioned, specific labels are stacked directly underneath it. These labels document exactly where the bug was found, the ecology of the area, and who identified it, because without this data, a specimen is essentially useless to science.
The final result is a true work of art. You might have a tiny insect glued to a paper point, or a perfectly mounted beetle. What's absolutely crucial are those neat,
perfectly aligned, readable labels stacked beneath them. This painstaking preparation ensures the specimen is ready to be safely stored in a research collection or museum, like the Smithsonian, for decades, allowing scientists to study changes in biodiversity over time.